Welcome, friend. As you settle into this quiet moment, let your breathing slow and your mind ease into a gentle rhythm. Tonight, we're going to explore something remarkable together. A story written in stone, in DNA, in the very architecture of living things. We're going to discover why, after more than a century and a half of intense scrutiny from thousands of scientists across every continent, the theory of evolution by natural selection stands stronger than ever before. This isn't about controversy or conflict. This is about evidence, mountains of it, oceans of it, gathered patiently over decades by researchers who dedicated their lives to understanding the truth about our natural world. As you listen, let the weight of this evidence wash over you like a warm tide, each fact settling comfortably into place. There's something deeply peaceful about certainty built on solid ground, about understanding that's been tested and refined and proven true time and time again. So relax, close your eyes if you haven't already, and let's begin this journey through time, through the fossil record, through the intricate code of life itself, the bedrock. What evolution actually is. Before we can understand why evolution has never been disproved, we need to understand what evolution actually means. And here's where we find our first point of clarity. Because evolution is often misunderstood, sometimes deliberately so, sometimes simply through honest confusion. Evolution at its most fundamental level means descent with modification. Picture a family tree stretching back through time. Your parents gave you certain traits, perhaps your mother's eyes, your father's height. But you're not identical to either of them. You're a modification of what came before. Now extend this principle across not just generations but across thousands, millions, even billions of generations. Extend it across not just your family, but across all families of all living things. That's evolution, simple in concept, profound in implication. Charles Darwin, that gentle naturalist who sailed around the world on the HMS Beagle in the 1830s, understood this principle better than anyone of his time. He observed finches on the Galapagos Islands, each species with beaks shaped differently, each perfectly suited to eating different food. He saw giant tortoises, whose shell shapes varied from island to island. He examined fossils of extinct creatures that resembled living species but weren't quite the same. And slowly, carefully, he pieced together an understanding that would transform biology forever. Darwin proposed that all living things share common ancestors. The birds and the reptiles, he suggested, descended from earlier forms that were neither quite bird nor quite reptile. The mammals and the reptiles before them descended from even earlier ancestors. Go back far enough, and you'd find that all vertebrates, animals with backbones, share a common ancestor. Go back further still, and all animals share ancestors. Further yet, and plants and animals diverge from a common origin. All the way back, some 3.5 billion years ago, to the first simple cells that could copy themselves. This might sound like speculation, but here's what makes it science rather than mere imagination. Evolution makes prediction. It says that if all life shares common ancestors, then we should find certain patterns in nature. We should find similarities in the basic biochemistry of all living things. We should find intermediate forms in the fossil record. We should be able to organize species into groups based on shared characteristics, and those groups should make sense in terms of which species are more closely related. We should see DNA similarities between closely related species and differences between distant relatives. We should observe small-scale evolutionary changes happening in real time. And remarkably, wonderfully, every single one of these predictions has been confirmed. Not once, not twice, but thousands upon thousands of times, in laboratories and fossil beds and genetic sequencing centers around the world. But evolution is more than just common ancestry. It's also about the mechanism that drives change over time. Darwin called this mechanism natural selection, and it's elegantly simple. In any population of living things, individuals vary. Some rabbits run faster than others. Some bacteria resist heat better than their neighbors. Some plants produce more seed. These variations exist because of tiny differences in genetic makeup. Mutations that arise randomly. Sexual reproduction that mixes genes in new combinations. Now here's the key. When the environment changes or when new challenges arise, some of these variations help survival and reproduction more than others. The faster rabbits escape predators more often. The heat-resistant bacteria survive when temperatures rise. The plants with more seeds leave more offspring. Over time, the traits that help survival and reproduction become more common in the population. This is selection, not conscious selection by any designer, but the automatic, inevitable result of some individuals leaving more offspring than others. Given enough time, natural selection can produce remarkable changes. A population of small shrew-like mammals can eventually give rise to whales, to bats, to elephants, to humans. Not because any individual animal transforms during its lifetime. That's not how it works. But because over millions of generations tiny changes accumulate, branch points occur, and populations diverge into new species. The timescales involved are vast, almost beyond comprehension. Earth formed about 4.6 billion years ago. The oldest fossils we've found, simple bacterial cells, date back 3.5 billion years. Complex animals didn't appear until about 600 million years ago. Dinosaurs ruled for about 105 million years before their extinction 66 million years ago. Modern humans emerged only about 300,000 years ago, a mere eye blink in geological time. These vast timescales are crucial to understanding evolution. Critics sometimes say, "I've never seen one species change into another." Of course not. Such changes take hundreds of thousands or millions of years, but we can see smaller evolutionary changes happening right now. In our own lifetimes, bacteria evolving resistance to antibiotics, insects adapting to pesticides, viruses mutating to evade our immune system. These are evolution in action, small steps in the endless journey of life, adapting to its environment. Now, a word about what evolution is not. Evolution is not about the origin of life itself. That's a different question, one that scientists are still working to answer. Evolution begins once you have reproducing organisms, once life exists, and can pass on traits to offspring. How the very first self-replicating molecules arose is abiogenesis, not evolution, though the two are related fields of study. Evolution is also not random. Natural selection is the opposite of random. It systematically preserves what works and eliminates what doesn't. Yes, mutations are random. Genetic variation arises by chance. But the selection process is anything but random. It's driven by the harsh but consistent logic of what allows organisms to survive and reproduce in their particular environment. Evolution is not about progress towards some goal. There's no ladder of complexity with humans at the top. Evolution has no foresight, no plan, no destination. It's simply populations changing in response to their environments. Sometimes that leads to greater complexity. Sometimes it leads to simplification. Parasites often lose organs their free-living ancestors had, because those organs became unnecessary. That's evolution too, losing features that no longer provide advantage. And perhaps most importantly, evolution is not just a theory in the way that phrase is commonly used. In science, a theory isn't a guess or a hunch. Atomic theory, germ theory, the theory of gravity. These are our most robust explanations of how nature works, their frameworks supported by mountains of evidence, tested and retested, refined over decades or centuries. The theory of evolution stands among these giants of science, supported by evidence from paleontology, comparative anatomy, embryology, molecular biology, biogeography, and direct observation. It's one of the most thoroughly documented phenomena in all of science. So that's what evolution is. Descent with modification through natural selection, operating over vast timescales, producing the diversity of life we see around us. It's a fact that species change over time. We've observed it directly. It's a theory that explains how and why this happens through natural selection, acting on heritable variation. The theory has been tested in every way imaginable, and it has passed every test. Now let's look at the evidence. Let's see why evolution hasn't just survived scrutiny. It's grown stronger with every new discovery. The fossil record. Ancient whispers from stone. Imagine you could open a book that tells the story of life on Earth, written in stone, preserved in layers of rock that formed over hundreds of millions of years. This book exists. We call it the fossil record, and it's one of the most powerful testimonies to the reality of evolution. Fossils are the preserved remains or traces of ancient organisms. Usually, when something dies, it decays quickly, scattered by scavengers, broken down by bacteria. But occasionally, very occasionally, an organism dies in just the right place, perhaps covered quickly by mud in a river, or buried by volcanic ash, or trapped in amber or ice. Under these special conditions, hard parts like shells, bones, or teeth can be preserved as minerals slowly replace the original material, or as impressions left in hardening rock. The chances of any individual organism becoming a fossil are vanishingly small. The chances of that fossil surviving millions of years of geological upheaval, not being destroyed by heat or pressure or erosion, and then being found by humans at or near the Earth's surface. Smaller still, yet we have found millions of fossils representing thousands of extinct species. The fossil record is incomplete. Certainly, we'll never have fossils of every species that ever lived. But the fossils we do have tell an extraordinarily coherent story. Here's what makes the fossil record such powerful evidence for evolution. The order of fossils matches exactly what evolutionary theory predicts. The deepest, oldest rocks contain only the simplest organisms, bacteria and single-celled creatures. No complex animals, no plants, just microscopic life. As you move up through the geological layers, through younger and younger rocks, complexity increases. First come simple multicellular organisms, then come creatures with hard shells and exoskeletons, then fish, then amphibians, then reptiles, then mammals and birds, then modern species. This order is consistent worldwide. Nowhere on Earth do you find mammals in the same rocks as trilobites. Nowhere do you find human fossils alongside dinosaurs, Nowhere do you find modern birds in rocks older than dinosaurs. The sequence is always the same. Simple to complex, ancient forms to modern forms. This pattern makes sense only if later species descended from earlier ones. Moreover, fossils within each layer can be grouped by similarity into families and orders, just as we group living species. Ancient fish in 400 million-year-old rocks share certain features: backbones, scales, fins. Ancient amphibians in 360 million-year-old rocks share different features: 4 limbs, different skull structures. Ancient reptiles in 300 million-year-old rocks show yet other characteristics. Each group appears after their ancestors and before their descendants, exactly as evolution predicts. But the fossil record offers more than just this consistent sequence. It offers something even more compelling: transitional fossils, organisms that show intermediate characteristics between major groups. Consider the evolution of whales, one of the most beautifully documented transitions in the fossil record. Modern whales are mammals. They breathe air, nurse their young, have hair, yet they live entirely in water and look nothing like land mammals. For many years, the evolutionary path from land mammals to whales was unclear. Then, starting in the 1980s, paleontologists working in Pakistan and Egypt began finding remarkable fossils that filled in the gaps. They found Pakicetus, a 50-million-year-old creature about the size of a wolf, with 4 legs clearly built for running on land. But Pakicetus had something unusual: ear bones structured like those of whales, different from all other land mammals. It likely spent time in shallow water, perhaps ambushing prey like a crocodile, but it lived primarily on land. Then there was Ambulocetus, meaning walking whale, from slightly younger rocks. Ambulocetus had powerful legs for swimming, nostrils farther back on its snout than Pakicetus, and a tail that could have helped propel it through water. It was amphibious, equally at home in water and on land. Moving forward in time, we find Rhodocetus, with even smaller hind legs and a more streamlined body for swimming. Then Dorodon and Basilosaurus, fully aquatic whales with tiny vestigial hind limbs still present but no longer functional for walking. Finally, we reach modern whales, which have lost their external hind limbs entirely, though tiny remnant bones remain buried deep in their bodies. This progression is exactly what evolution predicts: not sudden jumps from land mammal to whale, but a series of incremental changes, each species slightly different from its predecessor, moving gradually from land to water over millions of years. And we can date these fossils with radiometric methods, confirming that each intermediate form lived at the right time in the sequence. Or consider Tiktaalik, discovered in Arctic Canada in rocks 30. Before Tiktaalik was found, there was a gap in our understanding of how fish evolved into the first 4-limbed animals, the tetrapods. Scientists predicted that an intermediate form should exist in rocks of a certain age, in certain environment. They searched for years in rocks meeting those criteria, and in 2004, they found exactly what they were looking for. Tiktaalik had the body and scales of a fish, with gills for breathing underwater, but it also had a flat head with eyes on top like a crocodile, a flexible neck, and most remarkably, fins with bones arranged like the arm bones of tetrapods. The same pattern of one bone connected to 2 bones connected to many smaller bones that forms the arms and legs of all 4-limbed vertebrates, including Tiktaalik could do push-ups on its front fins, supporting its body weight, likely living in shallow water and occasionally venturing onto land. Before Tiktaalik was found, some critics claimed that the transition from fish to land animals was impossible. that no intermediate forms exist, yet evolution predicted such forms should exist. Scientists searched in the right rocks, and they found them. This is how science works, making predictions and testing them. The fossil record is full of such transition. We have fossils showing the gradual evolution of modern horses from small multi-toed ancestors. We have feathered dinosaurs showing the transition to birds, including specimens with both reptilian teeth and skeletal features alongside bird-like feathers and flight-related adaptation. We have fossils of early mammals showing the gradual evolution of specialized teeth, 3 middle ear bones and other mammalian features from reptilian ancestors. Now some ask, if evolution is true, why aren't there fossils for every single step in every evolutionary transition? This question misunderstands both the rarity of fossilization and the nature of scientific evidence. Fossilization requires specific conditions, quick burial, the right minerals, millions of years without erosion or destruction. Most organisms don't fossil. Most fossils that do form are destroyed by geological processes. Most fossils that survive are never found. They're buried too deep or in places we haven't looked. Despite these obstacles, we have thousands of transitional fossils. The fossil record is like a movie of life's history, with many frames missing, but enough remaining to tell the story clearly. Moreover, evolution doesn't require that every species leave fossils. What matters is the pattern we do see, and that pattern consistently supports evolutionary theory. The sequence of fossils, the timing of transitions, the geographical distribution, the nested hierarchy of traits, all point to descent with modification over vast time periods. Perhaps most tellingly, the fossil record has never contradicted evolution. If we found a rabbit fossil in Cambrian rocks 500 million years old, evolution would be in serious trouble. If we found human fossils alongside trilobites, evolution would be refuted. If fossils were randomly mixed throughout geological time with no consistent pattern, evolution would be falsified. But in the century and a half since Darwin published his theory, with millions of fossils collected from around the world, not one has contradicted the evolutionary sequence. Not one has appeared in the wrong time period. Every new fossil fits into the pattern predicted by evolutionary theory. This isn't proof. Science doesn't deal in absolute proof, but it's an overwhelming accumulation of evidence. The fossil record shows exactly what we'd expect if evolution were true, a consistent sequence from simple to complex, earlier to late, with transitional forms bridging the gaps between major groups, and with more transitional fossils being discovered every year, as paleontologists continue their work. As you lie here in the darkness, think about those ancient stones, scattered across continents, buried in deserts, and mountains and former sea floors, each one a snapshot of a moment in life's long journey, each one a piece of the puzzle. And when you put them together, they form a picture that tells the same story from every angle. Life has evolved over billions of years, from simple beginnings to the magnificent diversity we see today. Written in our cells, the DNA evidence. If the fossil record is a history book written in stone, then DNA is an archive stored in every living cell, a molecular document that records the relationships between all organisms with startling precision. And what this molecular evidence reveals is perhaps the most powerful confirmation of evolution we have. Every living thing on Earth, every bacterium, every fungus, every plant, every animal, stores its genetic information in DNA molecules. These long, twisted chains of chemicals spell out instructions for building and maintaining life using a 4-letter alphabet, A, T, G, and C, representing the 4 chemical bases that make up DNA.
The sequence of these letters determines what proteins get made, and proteins do virtually all the work in cells. Here's what's remarkable. The basic mechanism is the same in every organism. The genetic code that translates DNA sequences into proteins is universal across all life. Same three-letter DNA sequence codes for the same amino acid, whether you're looking at a bacterium, a rose, or an elephant. This universality makes sense if all life descended from a common ancestor that used this genetic code, and all descendants inherited it. It makes no sense if every species were created separately. Why would independent creation produce identical genetic systems across all living things? DNA allows us to do something fossils cannot. We can directly compare the genetic sequences of different species to determine how closely related they are. The principle is straightforward. When a population splits into two separate populations that no longer interbreed, they begin accumulating different mutations in their DNA. The longer they've been separated, the more differences accumulate. Closely related species, which diverged recently, have very similar DNA. Distantly related species, which diverged long ago, have more difference.
This molecular clock is remarkably consistent. Humans and chimpanzees, our closest living relatives, share about 98.8% of our DNA sequence. We split from our common ancestor only 6 to 7 million years ago. Humans and gorillas share slightly less, about 98.4%. Because we split from gorillas a bit earlier, humans and orangutans share less. Still, humans and Old World monkeys share even less, because that split happened about 25 million years ago. The pattern continues outward. We share less with other mammals, still less with reptiles, even less with fish, and so on. But here's what's crucial. The pattern of similarity in DNA matches exactly the pattern predicted from comparative anatomy, embryology, and the fossil record. The organisms that look most similar anatomically also have the most similar DNA. The order of divergence suggested by fossils matches the order suggested by DNA differences. Three completely different types of evidence, bones, genes, and rocks, all tell the same story. This consilience, this coming together of independent lines of evidence, is powerful confirmation that we're understanding reality correctly. DNA reveals relationships that we couldn't have guessed from anatomy alone. For instance, traditional classification put whales in their own order, separate from hoofed mammals. But DNA analysis revealed that whales are actually most closely related to hippos. Both descended from a common ancestor about 55 million years ago. This finding, initially surprising, led paleontologists to look for specific skeletal features that whales and hippos might share. And they found them. The molecular evidence guided the anatomical research. But DNA offers more than just a family tree. It also provides evidence of evolution's history in unexpected ways. Consider pseudogene sequences in our genome that look like genes but no longer function. They're like broken machinery in the genetic toolkit, relics of genes that were once functional but have accumulated mutations that rendered them useless. For example, most mammals can make their own vitamin C. The gene for the enzyme that produces vitamin C is active and functional in these species, but humans, other primates, and guinea pigs have lost this ability. We must get vitamin C from our diet or we develop scurvy. We still have the gene for the vitamin C making enzyme, but it's broken and inactivated by a mutation. It's a pseudogene. Here's what's telling.
We can trace the exact mutation that broke this gene, and that same mutation is found in all primates. The gene became non-functional in our common ancestor, and all descendants inherited the broken version. Different species have different additional mutations in this pseudogene, accumulated since they diverged, but they all share that original breakpoint. This pattern only makes sense if we share common ancestry with other primates. If we were created independently, why would we share the exact same broken gene? There are thousands of pseudogenes in the human genome, and we can trace their history, comparing how they've changed across species, mapping the tree of relationships. The patterns always support common ancestry. Or consider endogenous retroviruses, viral DNA sequences that got inserted into the genome of an ancestor and were passed down to descendants. When a retrovirus infects a cell, it inserts its genetic material into that cell's DNA. Usually this affects only that one cell, but occasionally a retrovirus infects a sperm or egg cell, and then its DNA becomes part of the organism's heritable genome, passed to offspring. Humans have thousands of these endogenous retroviruses in our genome, making up about 8% of our DNA. Most are ancient, degraded, no longer functional, but we can still recognize them as viral sequences. And here's the key. We share many of these viral insertions with other primates. Humans and chimpanzees have endogenous retroviruses in exactly the same positions in their genomes, the same viral sequence inserted in the same spot between the same surrounding genes. The odds of this happening by coincidence are astronomically small. Each viral insertion is essentially random, landing anywhere in 3 billion base pairs of DNA. For two species to have the same insertion in the same spot independently would be like two people randomly typing sequences of 3 billion letters and happening to insert the same sentence in the exact same position. It doesn't happen by chance. It happens through inheritance from a common ancestor who had that viral insertion. DNA also reveals how evolutionary changes occur at the molecular level. We can see gene duplications where a gene gets copied, allowing one copy to mutate and potentially develop a new function while the other copy maintains the original function. We can track how regulatory genes that control when and where other genes are active, have changed, producing major evolutionary innovations. We can observe how chromosomes have broken and rejoined over evolutionary time. Humans, for example, have 46 chromosomes, while our closest relatives, chimpanzees and gorillas, have 48. Did we gain chromosomes or did they? DNA analysis reveals the answer. Human chromosome 2 is the result of two ancestral chromosomes fusing together. We can still see the telomeres, the specialized sequences that normally appear only at chromosome ends right in the middle of human chromosome 2, where two ancestral chromosomes join. We can see remnants of what used to be a centromere, the structure that helps chromosomes divide properly, also in the middle of human chromosome 2. This fusion happened in a human ancestor after we split from the chimpanzee lineage. The precision of DNA evidence is stunning. We're not relying on impressions or judgments about similarity. We're comparing billions of base pairs letter by letter, measuring exact percentages of difference, calculating rates of change, constructing phylogenetic trees using sophisticated statistical methods, and the results overwhelmingly confirm evolution. Moreover, DNA evidence is testable and falsifiable. If evolution were false, we should find genetic sequences that don't fit the predicted patterns. We should find species that group differently, based on different. We should find DNA similarities that contradict the relationship suggested by anatomy or fossils. But we don't. Every new genome sequence, every new gene compared, adds to the consistent picture of life's evolutionary truth. Geneticist Theodosius Dobzhansky once wrote, "Nothing in biology makes sense except in the light of evolution." This is nowhere more apparent than in molecular biology. The patterns in DNA, the shared sequences, the pseudogenes, the endogenous retroviruses, the chromosomal rearrangements, all of these make perfect sense as products of descent, with modification from common ancestors. They make no sense any other way. As you drift towards sleep, picture those long spirals of DNA coiled in every cell of your body. Picture the same basic structure in every living thing, all using the same code, all showing patterns of similarity that map exactly onto the evolutionary tree, constructed from completely independent evidence. This isn't just one line of evidence, it's millions of lines, each genetic comparison another confirmation, another piece fitting perfectly into place. The molecular evidence alone would be enough to establish evolution beyond reasonable doubt. Combined with everything else, it's overwhelming. The architecture of life, comparative anatomy. There's something quietly beautiful about looking at the bones of different animals and seeing beneath the surface differences, the same fundamental structures playing variations on a theme. A human arm, a cat's front leg, a whale's flipper, a bird's wing, a bat's wing. At first glance, these look nothing alike, but strip away the flesh and muscle, lay out the skeletons, and something remarkable appears. The same bones, arranged in the same order, modified for different purposes. One bone in the upper arm, two bones in the lower arm, a cluster of small wrist bones, 5 digits. This is the basic pentadactyl, 5-fingered limb pattern, shared by all tetrapods, all 4-limbed vertebrates. In humans, those 5 digits are fingers and thumbs, perfect for grasping and manipulating tools. In a cat, they're paw bones, with claws for catching prey. In a whale's flipper, they're embedded in a paddle-shaped structure for swimming. In a bird's wing, some are fused and elongated to support feathers for flight. In a bat's wing, they're stretched to enormous lengths, with thin skin membranes between them for powered flight. Different functions, different appearances, but the same underlying architecture. Why would this? If each species were designed separately for its specific needs, why give a whale finger bones inside its flipper? Why not design something completely different, optimized for swimming, with no trace of digits? Why give a bird a hand with fingers inside its wing? The answer makes sense only through evolution. These animals all inherited the basic limb pattern from a common ancestor, and natural selection modified that ancestral pattern in different ways to suit different lifestyles. You can't redesign from scratch. Evolution works with what's already there, tinkering with existing structures, not creating entirely new ones. The result is unity of pattern beneath diversity of form. This is what's called homology, similarity due to shared ancestry, and it's everywhere in biology. Consider hearts. Fish have 2 chambers in their hearts. Amphibians typically have 3. Reptiles have 3 or 4 depending on the type. Birds and mammals have 4. You can trace the evolutionary development of the heart through these increasing complexities, each change representing an adaptation to different lifestyles and different ways of getting oxygen to tissue.
Or look at vertebrate embryos, fish, amphibians, reptiles, birds, mammals. Despite looking radically different as adults, their embryos are remarkably similar in early development. All vertebrate embryos, including human embryos, have pharyngeal arches, structures that look like gill slits. In fish, these develop into actual gills. In mammals, including humans, they develop into the lower jaw, the bones of the middle ear, and other structures of the face and neck. Why would human embryos have structures that look like gills? Because we evolved from ancestors that had gills, and we've retained those embryonic structures, repurposed for new functions as development proceeds. These similarities in embryological development were recognized even before Darwin, but Darwin's theory explained them. Embryos show similarities because they reflect shared evolutionary history. The earlier the developmental stage, the more similarities you see across different species, because those early stages evolved long ago in common ancestors. Then there are vestigial structures, features that are reduced, rudimentary versions of structures that were fully functional in ancestors. These are some of the most direct evidence of evolution, because they only make sense as evolutionary remnants. Human tailbones, for example. We have a coccyx, the remnant of a tail, complete with the same vertebrae that form tails in other mammals. It serves no function beyond being an anchor point for a few muscles, certainly not the function of a proper tail. But our mammalian ancestors had tails, and we've retained the skeletal remnants. Or consider the human appendix, that small pouch attached to the large intestine. In humans it has minimal function, perhaps some role in immune system development, but in many herbivorous animals, the cecum, of which the appendix is a remnant, is large and important for digesting plant material. Our primate ancestors likely had a functional cecum. As our lineage evolved toward a more varied diet, the cecum shrank, leaving us with the vestigial appendix. Whales and some snakes have pelvic bones and tiny leg bones buried deep in their bodies, completely useless for locomotion. Why would an aquatic mammal have hip bones? Why would a snake that has no legs have leg bones? Because their ancestors had legs. These are evolutionary leftovers, structures that have atrophied over millions of years but haven't completely disappeared. Some plants have flowers but never produce fruit. Some cave-dwelling fish have eye structures but are blind, the eyes covered by skin. Flightless birds like ostriches have wings that can't support flight. These vestigial structures are predicted by evolution and explained by evolution. They're remnants of once functional features preserved in reduced form. Comparative anatomy also reveals convergent evolution, when unrelated organisms evolve similar features independently, because they face similar challenges. The camera-like eyes of vertebrates and octopuses, for instance, look remarkably similar and work on the same principles, but they evolved independent. We know this because the internal structure differs. Octopus eyes don't have the blind spot that vertebrate eyes have, and they develop from different tissue in embryos. Similar function, similar structure, but different evolutionary origin. Or consider the wings of birds, bats, and insects, all used for flight, all wing-shaped, but fundamentally different in construction. Bird wings are modified arms with feathers. Bat wings are also modified arms but with skin stretched between elongated fingers. Insect wings are outgrowths of the exoskeleton, with no bones at all. Same function, different structures, different evolutionary paths to solving the same problem. This is how we distinguish homology from analogy. Homologous structures share common ancestry, like the pentadactyl limb pattern. Analogous structures serve similar functions but have different origins, like wings across different animal groups. Understanding this distinction helps us trace evolutionary relationships accurately. The patterns revealed by comparative anatomy are stunningly consistent with the predictions of evolutionary theory. The nested hierarchy of traits, some shared by all vertebrates, others shared only by mammals, still others shared only by primates, creates a family tree that matches perfectly with the trees created from DNA evidence and from the fossil record. Three independent approaches converging on the same answer. Moreover, comparative anatomy has predictive power. Scientists can predict what features an extinct animal had based on its relationships to living species. They can predict where to find certain bones or organs in species they've never examined, based on patterns seen in related species. These predictions are consistently confirmed. No alternative explanation accounts for these patterns as completely as evolution. Why would separately created species share vestigial organs? Why would they share the same bone patterns used for wildly different purposes? Why would embryos of different species look similar and then diverge? Why would the patterns of similarity form nested hierarchies, matching independent molecular and fossil evidence? Evolution explains all of it. Homologous structures reveal shared ancestry. Vestigial organs show evolutionary history. Analogous structures demonstrate how different lineages solve similar problems. The tree of relationships constructed from anatomy matches the tree from DNA and fossil. The evidence interweaves, each strand supporting the others, creating a picture of life's history that grows clearer with every new discovery. As you settle deeper into rest, imagine the elegant economy of evolution, working with existing structures, modifying them, repurposing them, sometimes letting them atrophy but rarely discarding them entirely. Imagine the long chain of ancestors, each generation passing on its basic body plan, with slight modifications, those modifications accumulating over vast time, until a land mammal's leg becomes a whale's flipper, or a reptile's grasping forelimb becomes a bird's wing. The patterns are there in the bones, waiting to be read by those who take time to look. Islands of evidence, biogeography and distribution. If you were to invent species independently, placing them around the world, you'd probably put each type of organism in every environment where it could thrive. You'd put lions on every continent with suitable grass. You'd put penguins in Arctic waters as well as Antarctic. You distribute tropical plants evenly across all equatorial regions. But that's not what we find in nature. Instead, we find a very different pattern, one that makes perfect sense in light of evolution and continental drift, but no sense otherwise. Take Australia, that vast island continent that separated from other landmasses about 40 million years ago. Australia has no native placental mammals except for bats and rodents that could fly or raft across water. Instead, it's dominated by marsupials, mammals that carry their young in pouches. Australia has marsupial mice, marsupial wolves, marsupial moles, marsupial flying squirrels, and dozens of other forms, each filling ecological roles that placental mammals fill on other continents. Why would Australia be so different? Evolution explains it clearly. When Australia separated from other continents, it already had marsupials but few placentals. Isolated for millions of years, those marsupials diversified into many forms, adapting to different niches, while placentals diversified separately on other continents. Each landmass developed its own distinctive fauna based on what ancestors were present when isolation began. This pattern appears everywhere. Madagascar, isolated for about 90 million years,
has lemurs found nowhere else on Earth. South America, isolated for much of the past 60 million years, developed unique mammals including armadillos, anteaters, and sloths. When Central America formed a land bridge between North and South America about 3 million years ago, species from both continents, many South American species went extinct in competition with northern mammals, and we saw the Great American Interchange, a natural experiment in biogeography that played out exactly as evolutionary theory would predict. Islands are particularly instructive. Oceanic islands that formed from volcanic activity and never connected to continents show a distinctive pattern. They have relatively few species, often with unusual characteristics, and many endemic species found nowhere else. They also notably lack certain groups of animals that would thrive there if they could reach it. The Hawaiian Islands, for example, have no native amphibians, no native land mammals except a bat, no native terrestrial reptile, yet they have an incredible diversity of honey creeper birds, more than 50 species, all descended from a single finch-like ancestor that reached the islands and then diversified into forms with different beaks, different feeding habits, different lifestyle. This adaptive radiation is exactly what evolution predicts. A colonizing species, finding empty niches, diversifies to fill those niches over time. The Galapagos Islands, where Darwin observed those famous finches, show the same pattern. The finches on Galapagos all descended from a common ancestor from mainland South America. Once isolated on different islands, populations diverged, adapting to different food sources. Some developed thick beaks for cracking hard seeds, others developed thin pointed beaks for eating insects. Still others developed beaks for feeding on cactus flowers, or even for using tools to extract insect larvae from wood. Each variation arose through natural selection, favoring individuals best suited to utilize available resources. Why don't Galapagos Islands have frogs or salamanders? They're volcanic islands that rose from the ocean floor and never connected to a continent. Amphibians can't cross wide stretches of salt water. They never reached the islands, so they never evolved there. But finches, which can fly long distances, did reach the islands, and they did evolve there. The presence and absence of species on islands makes sense only through the lens of evolutionary history and biogeography.
Continental distributions tell similar stories. Why are there no elephants in South America, even though suitable habitat exists? Because elephants evolved in Africa and Asia, and they never reached South America. Why are there camels in Africa, Asia, and Australia, but not in South America, even though South America has llamas and alpacas? Because camels and llamas share a common ancestor that lived in North America. One branch of descendants migrated across the Bering Land Bridge to Asia, eventually reaching Africa. Another branch stayed in the Americas, eventually evolving into llamas and alpacas. Then most of the North American populations went extinct, which is why we find the descendants scattered across distant continents rather than in their ancestral homeland. Freshwater fish distributions provide more evidence. Fish in different rivers on the same continent are often closely related, even when the rivers are now separate, because those rivers were connected during past geological periods. Fish on different continents, even when living in similar environments, are usually quite different, because they evolved separately after continental separation. The pattern reflects history, not just habitat. Plant distributions show the same biogeographical pattern. Similar environments on different continents have different plant communities, because different ancestors were present when continents separated. The African savanna looks different from Australian grasslands or South American pampas, even though all are tropical grasslands, because each has a distinct evolutionary history. Perhaps most telling are the relationships between organisms on oceanic islands and their nearest continental relatives. Hawaiian honeycreepers are most closely related to birds from the Americas. Galapagos finches are most closely related to South American birds. Madagascar's lemurs are most closely related to African primates. This pattern makes sense if species colonized these islands from nearby continents and then evolved in isolation. It makes no sense if species were independently placed. The fossil record adds another dimension to biogeography. We find fossils of species in locations that seem wrong by modern standards, but make sense given past continental positions. Fossils of similar species on now widely separated continents, like matching Mesosaurus fossils in South America and Africa, helped convince geologists that continents had once been joined. The distribution of fossils traces the movement of continents over geological time. Modern molecular techniques let us test biogeographical hypotheses with precision. We can determine when different populations diverged by comparing their DNA, and those dates match up with geological evidence of when continents separated or when land bridges formed or flooded. The molecular clock ticks in harmony with the geological clock. All of this, the unique faunas of isolated continents, the endemic species on islands, the absence of certain groups where we'd expect them if species were independently distributed, the relationships between island and continental species, all of it makes perfect sense as the product of evolution in geographically isolated populations. Species originate in one location and spread from there, diverging as populations separate and adapt to new environments. Barriers, oceans, mountains, climate zones, limit which species can reach which areas. Time and isolation allow divergence and speciation. This is biogeography's contribution to the case for evolution. The distribution of species across the planet reflects historical patterns of descent, dispersal, and adaptation, not optimal placement for each environment. It tells a story of ancestry and migration, of populations splitting and diverging, of continents drifting and barriers forming. And this story matches perfectly with the story told by fossils, by DNA, by comparative anatomy. Different types of evidence, independent methods. Same conclusion. As you rest here, think about those island finches, those Australian marsupials, those Hawaiian honeycreepers, each group telling the same essential story. Life diversifies when populations separate and adapt to their local conditions. The geography of life is the geography of evolutionary history, mapped across the globe, written in the patterns of where species are found and where they're absent, in who's related to whom, in how distributions change over time as continents shift and climates alter. It's evidence of such clarity that once you see the pattern, it's impossible to unsee. Evolution before our eyes, observable changes. One of the most common misconceptions about evolution is that it's impossible to observe because it happens too slowly. And it's true that major transitions, fish to amphibians, reptiles to birds, land mammals to whales, require millions of years. But evolution itself, the process of populations changing over time through natural selection, happens constantly. We can watch it occur, we can measure it, we can even predict it. Consider antibiotic resistance in bacteria. This is evolution happening in real time right now, in hospitals and clinics around the world. When antibiotics were first introduced in the 1940s, they were remarkably effective. Bacterial infections that once killed millions
could be cured with a simple injection, but over time, something changed. Bacteria evolved. Here's how it happened. In any large population of bacteria, random mutations create genetic variation. Most mutations have no effect or are harmful, but occasionally a mutation gives a bacterium some resistance to an antibiotic, perhaps by changing a protein that the antibiotic targets, or by producing an enzyme that breaks down the antibiotic, or by altering the bacterial membrane so the antibiotic can't enter. When you take antibiotics, you create strong selective pressure. Bacteria without resistance die. Bacteria with even slight resistance survive. They reproduce, passing the resistance gene to their offspring. With each generation, resistance becomes more common in the population. After multiple rounds of antibiotic exposure, what was once a rare trait becomes standard. The population has evolved. This isn't theoretical. We've documented it thousands of times. Methicillin-resistant Staphylococcus aureus, MRS. Now it causes hundreds of thousands of infections yearly. Tuberculosis strains resistant to multiple drugs have emerged. New resistance mechanisms appear in bacteria with such regularity that pharmaceutical companies struggle to keep developing new antibiotics faster than bacteria evolve. Resistance to old ones. The same principle applies to pesticide resistance in insects. When DDT was first used as an insecticide in the 1940s, it was devastatingly effective, but within a few years, resistant flies appeared. By the 1960s, resistance was widespread. The same pattern has repeated with other pesticides. Initial effectiveness, then resistance emerges and spreads. Houseflies today can resist pesticides that would have killed their great-grandparents instantly. Or consider the peppered moth in England, one of the most famous examples of natural selection observed in nature. Before the Industrial Revolution, peppered moths were typically light-colored, camouflaged against light-colored tree bark, covered with lichens. Darker moths existed, but were rare because they were easily spotted and eaten by birds. Then industrialization brought pollution that killed the lichens and darkened the trees with soot. Now the light moths stood out and the dark moths were camouflaged. Within a few decades, the moth population shifted from mostly light to mostly dark. When pollution controls were implemented and trees became lighter again, the moth population shifted back toward lighter colors. The allele frequencies in the moth population tracked environmental changes over just a few generations. Darwin's finches on the Galapagos have been studied continuously since 1973 by Peter and Rosemary Grant and their colleagues. They've measured every finch on one island, tracking beak sizes, body sizes, survival rates, and environmental conditions year by year. They've watched evolution happen. During droughts, when only large, tough seeds are available, finches with larger, stronger beaks survive and reproduce better. The next generation has, on average, larger beaks. During wet years, when small, soft seeds are abundant, smaller beaks become advantageous. The average beak size in the population shifts back and forth, tracking environmental conditions. The changes are small year by year, but they're measurable, and they're exactly what natural selection predicts. Even more dramatically, researchers observed the formation of a new hybrid species when a large beaked finch from one island bred with a native finch on another island. The hybrids had intermediate traits and bred primarily with each other, not with either parent species, creating a reproductively isolated population, the beginning of a new species observed in real time. HIV provides another clear example. The virus evolves so rapidly that doctors must prescribe multiple drugs simultaneously to stay ahead of its evolution. Using a single drug creates selective pressure, and resistant viral variants emerge within weeks or months. But multiple drugs simultaneously raise the bar. Resistance to all drugs at once is much rarer. Even so, resistant strains sometimes evolve, demonstrating natural selection's power even within a single infected patient. Over the course of a few years, influenza viruses evolve so quickly that flu vaccines must be reformulated every year. The strain circulating this year is different enough from last year's strain that last year's vaccine won't fully protect you. Over decades, the influenza virus has diverged into multiple lineages, each adapting to its own hosts and conditions. Scientists can trace this viral evolution, mapping the relationships between strains, predicting which strains will become dominant next season based on their genetic changes and selective advantages. Laboratory experiments have demonstrated evolution under controlled conditions hundreds of times. Richard Lenski's E. coli long-term evolution experiment has been running since 1988, tracking 12 populations of bacteria over more than 7. The populations have evolved larger cell sizes, higher fitness, and new metabolic capabilities, including one population that evolved the ability to metabolize citrate, something the ancestral E. coli couldn't do. All these changes are documented, with frozen samples of each population stored every few hundred generations, so researchers can look back and trace exactly how and when changes occur. Lizards transplanted to new islands have evolved changes in leg length, body size, and jaw strength within just a few decades as they adapt to new environments. Guppies moved to streams with different predators, evolved different life history strategies, earlier or later reproduction, more or fewer offspring within a few years. Stickleback fish populations that colonized freshwater lakes from the ocean have repeatedly evolved similar adaptations to freshwater life, a natural experiment replicated many times in different lakes, showing that evolution is predictable when environments are similar. These observations destroy the argument that evolution is too slow to observe, or that it only happens in the past. We see populations changing in response to environmental pressures all the time. We can measure the changes. We can identify the genes responsible. We can predict when and how resistance will develop. We can watch species diverge. Evolution isn't just a historical inference. It's an ongoing process we observe directly. Now, skeptics might say, these are just small changes within existing species. You're not seeing new species form. But first, speciation doesn't happen overnight. The populations in Lenski's experiment or Grant's finch studies are in the process of diverging. Give them another million years with continued isolation, and they might well be different enough to call separate species. Second, we have observed speciation in plants through polyploidy, where chromosome doubling creates instant reproductive isolation. And third, these observed changes are exactly the raw material of evolution. The same processes that produce antibiotic resistance or different finch beaks, given enough time and accumulated changes, produce larger transformations.
The objection also misunderstands what evolutionary theory claims. Evolution doesn't say a bacterium will become a fungus, or an insect will become a bird. It says populations change over time through natural selection, acting on variation. Small changes accumulate. Populations diverge when isolated. Over vast time, this produces the diversity of life. What we observe in real time, populations adapting to environments, frequencies of traits changing, new capabilities emerging through genetic changes, is exactly what the theory predicts at small scale. The large-scale patterns we see in fossils, anatomy and DNA are exactly what we'd expect from these small-scale processes operating over millions of years. These contemporary observations of evolution aren't peripheral curiosities. They're central confirmations of evolutionary theory's core claims. Natural selection works. Populations evolve. Genetic changes spread through populations when they provide advantages. Environmental pressures shape the trajectory of evolutionary change. All the fundamental principles of evolution can be observed operating today. Let these examples settle into your mind as you drift towards sleep. Evolution isn't ancient history. It's happening now, in bacteria in our hospitals, in viruses in our bodies, in insects in our fields, in finches on remote islands. It's measurable, predictable, observable, and every observation confirms what Darwin proposed over 150 years ago. Life changes over time through natural selection, acting on heritable variation. The theory isn't speculation about the distant past. It's a description of processes operating all around us, every day shaping the living world continuously. The web of confirmation, multiple lines of evidence. Science at its best doesn't rely on a single line of evidence. It weaves together many independent strands, each adding strength, each capable of standing on its own, but far more powerful in combination. And this is exactly what we have with evolution: multiple, independent lines of evidence, all pointing to the same conclusion. The fossil record tells one story. Layer upon layer of rock preserves the sequence of life through time, showing simple organisms in old rocks, complex organisms in younger rocks, transitional forms bridging the gaps between major groups. This evidence alone would strongly suggest that life has evolved over billions of years. The genetic evidence tells another story. DNA sequences reveal patterns of similarity and difference that map species relationships, showing nested hierarchies of shared genes, pseudogenes marking common ancestry, endogenous retroviruses appearing in the same locations in related species. This evidence alone would convincingly demonstrate common descent. Comparative anatomy tells yet another story. Homologous structures reveal shared ancestry through similar bone patterns modified for different functions. Vestigial organs show evolutionary history through reduced remnants of once functional features. Embryological similarities trace back through development to common origins. This evidence alone would strongly indicate evolutionary relationships. Biogeography tells still another story. The distribution of species across continents and islands reflects patterns of descent and dispersal, not optimal placement. Endemic species on isolated islands, unique faunas on separated continents, relationships between island species and continental ancestors, all pointing to evolution in geographically isolated populations. Direct observation tells a final story. We watch evolution happen in bacteria, insects, viruses, finches, and countless other organisms. We measure populations changing. We see natural selection at work. We document speciation beginning. Now here's what's remarkable. These 5 completely independent lines of evidence all tell the same story. The relationships between species, suggested by fossils, match the relationships suggested by DNA. The tree of life constructed from comparative anatomy matches the tree from molecular biology. The timing of divergences estimated from genetics matches the timing shown in the fossil record. The biogeographical patterns match the patterns predicted from phylogenetic relationships. The processes we observe operating today, given enough time,
would produce exactly the patterns we see in the historical record. This consilience, this coming together of independent evidence, is extraordinarily powerful. Each type of evidence was developed using different methods, by different researchers, asking different questions. Paleontologists studying rocks had no access to DNA sequences. Molecular biologists sequencing genes weren't thinking about island biogeography. Anatomists examining bone structures weren't consulting fossil dates. Yet all their findings converge on the same answer. Life evolved over billions of years, through descent with modification, with populations diverging and adapting, driven by natural selection and other evolutionary processes. This isn't coincidence, it's confirmation. If evolution were false, these independent lines of evidence wouldn't all point the same direction. We might find fossils that contradicted DNA evidence. We might see biogeographical patterns that made no sense given anatomical relationship. We might observe genetic changes that couldn't produce the morphological changes we see in the fossil record, but we don't. Every new fossil fits into the predicted pattern. Every new genome sequence confirms the expected relationship. Every observed instance of evolution demonstrates the predicted mechanisms. The different types of evidence don't just coexist, they actively support each other, filling in gaps, testing predictions, providing mutual confirmation. Consider how this works in practice. Molecular data suggested that whales were closely related to hippos and other archaeodactyls, which was surprising given how different whales look. This led paleontologists to search for specific anatomical features that might link whales and archaeodactyls, and they found them particularly in the structure of the ankle bone. The molecular prediction was confirmed by anatomy. Then fossils of early whales were discovered showing the transition from land to sea, complete with intermediates having both terrestrial and aquatic features. Three types of evidence: molecules, bones and fossils, all telling the same story. Or consider human evolution. DNA showed that humans and chimpanzees diverged 6 to 7 million years ago. This predicted that hominid fossils should appear in rocks older than 6 million years, but not in rocks 10 or 20 million years old. And that's exactly what we find. The oldest hominid fossils date to around 6 to 7 million years ago. Older fossils don't show the characteristics that distinguish hominids from other apes. The fossil record matches the molecular prediction perfectly. The power of multiple independent lines of evidence is why scientists are so confident in evolutionary theory, despite never having witnessed the entire history of life. We don't need to see every step to know the story. When different types of evidence independently point to the same conclusion, and when that conclusion makes testable predictions that are repeatedly confirmed, we can be confident we're understanding reality correctly. This is how science builds reliable knowledge, not through single experiments or single observations, but through the accumulation of evidence from multiple sources, tested and retested, examined from every angle. Evolution has withstood more scrutiny than perhaps any other major scientific theory. It's being tested by specialists in dozens of different fields: paleontology, molecular biology, comparative anatomy, embryology, biogeography, genetics, population biology, systematics. Every field has added evidence. No field has refuted it. More than that, evolution has made predictions that were later confirmed. Darwin predicted that fossils of human ancestors should be found in Africa based on biogeography. A century later, that prediction was spectacularly confirmed when fossil hominids were discovered in Africa. Evolutionary theory predicted that we should find transitional fossils between fish and tetrapods in rocks of a certain age and type. Tiktaalik was found in exactly the right rocks in exactly the right time period. Genetic theory predicted that humans should have fewer chromosomes than other apes, if our chromosome 2 was a fusion. When we examined chromosomes, we found exactly that. These aren't retrofitted explanations, they're predictions made before the data was collected, then confirmed by observation. That's how robust scientific theories work. They don't just explain existing data, they predict new findings, and evolution has passed this test repeatedly.
The explanatory power of evolution is equally impressive. Why do species on isolated islands resemble nearby mainland species? Evolution explains it. Why do we have tailbones? Evolution explains it. Why do embryos of different species look similar? Evolution explains it. Why do bacteria become resistant to antibiotics? Evolution explains it. Why does DNA show nested hierarchies of similarity? Evolution explains it. Why do fossils appear in a consistent sequence? Evolution explains it. No competing explanation accounts for all these observations as successfully as evolution does. Some might explain one type of evidence but fail on others. Evolution explains all of them coherently, as aspects of a single unified process. As you lie here in the quiet darkness, let this realization sink in. The evidence for evolution isn't one or two findings that could be interpreted differently. It's thousands upon thousands of independent pieces of evidence, from dozens of scientific disciplines, all pointing the same direction, all fitting together like pieces of an enormous puzzle to reveal a single clear picture. This is why scientists accept evolution so completely. Not because of faith or ideology, but because the evidence is overwhelming, mutually reinforcing, and tested from every conceivable angle. The consilience of evidence creates a web of confirmation so strong that no single strand could break it, addressing the challenges. Why evolution stands strong. Despite the overwhelming evidence, evolution faces persistent challenges from those who reject it for religious, philosophical, or ideological reasons. It's worth examining these challenges, not because they cast doubt on evolution. They don't. But because understanding why they fail helps clarify why the evidence for evolution is so strong. Perhaps the most common objection is that evolution is just a theory. This betrays a fundamental misunderstanding of how science uses the word theory. In everyday speech, theory means a guess or speculation. In science, a theory is a comprehensive explanation of some aspect of nature that's been extensively tested and confirmed through observation and experimentation. We have the theory of gravity, which explains why objects fall and planets orbit. We have atomic theory, which explains the structure of matter. We have germ theory, which explains infectious disease. We have the theory of plate tectonics, which explains earthquakes and continental drift. And we have evolutionary theory, which explains the diversity and relationships of living things. These theories aren't guesses. They're our best, most thoroughly tested explanations of natural phenomena. Calling evolution just a theory is like saying gravity is just a theory, while falling objects somehow prove gravity wrong. The word theory in science denotes strength, not weakness. It means an explanation has survived rigorous testing and repeatedly been confirmed. Another common objection claims there are no transitional fossils, or that the gaps in the fossil record disprove evolution. But as we've seen, transitional fossils are abundant.
We have beautiful sequences showing the transition from fish to tetrapods, from land mammals to whales, from dinosaurs to birds, from early hominids to modern humans. Each year, paleontologists discover more. Yes, gaps exist. Fossilization is rare, and not every species leaves fossil. But the relevant question is, do the fossils we have fit the pattern evolution predicts? And the answer is an overwhelming yes. Fossils appear in the right sequence, with the right characteristics, at the right time. No fossils contradict evolutionary predictions. No rabbits in the Cambrian. No modern birds with dinosaurs, no mixing of time periods. The pattern is exactly what evolution predicts. The gaps argument also misunderstands evolution. Evolution doesn't say every species must leave fossils or that every transition must be perfectly preserved. It says species change over time and are related by common ancestry. The fossils we have amply demonstrate this, and the gaps that remain are gaps in our knowledge, not gaps in evolution itself. Some claim evolution has been disproved by complexity, particularly through the idea of irreducible complexity. The argument that certain biological structures are too complex to have evolved step by step because removing any part would make them non-functional. But this argument has been repeatedly refuted. First, many supposedly irreducibly complex structures have had their evolutionary pathways traced. Bacterial flagella, blood clotting cascade, the immune system, all examples cited as irreducibly complex, have been shown to have plausible evolutionary origins, often with simpler versions existing in other organisms or with components serving different functions that were later co-opted for new purpose. Second, the argument assumes that earlier versions of a structure must serve the same function as the final version. But evolution often repurposes existing structures. Feathers probably evolved for insulation or display before being used for parts of the eye existed for other functions before being assembled into the complex visual system of vertebrates. Evolution is opportunistic, cobbling together new functions from existing parts. Third, the irreducible complexity argument is essentially an argument from ignorance. I can't imagine how this evolved, therefore it didn't. But scientists can and have imagined and demonstrated evolutionary pathways for complex structure. The inability to immediately see how something evolved isn't evidence against evolution. It's a challenge to investigate further. And when scientists do investigate, they consistently find evolutionary explanation. Some argue that natural selection can't create new information or increase complexity. But this too has been refuted by direct observation. We've watched information increase through gene duplications. We've seen new metabolic capabilities evolve in bacteria. We've observed complexity increase through simulations and experiments. Lenski's E. coli evolved the ability to metabolize citrate, a new capability, new information, evolved through natural mechanisms. Others claim that evolution is unfalsifiable, that no observation could disprove it, making it unscientific. But this is false. Evolution makes many testable predictions, and any of them failing would challenge the theory. If we found fossils in the wrong sequence, evolution would be in trouble. If DNA didn't show patterns of nested similarity, evolution would be undermined. If we never observed natural selection producing adaptation, the mechanism would be in doubt. If comparative anatomy showed random patterns with no correlation to other data, evolution would be falsified. But these things don't happen. Every test supports evolution. Every new finding fits the predicted pattern. That's not unfalsifiability. That's robust confirmation. Some argue from personal incredulity. They simply can't believe complex organisms could arise without a design. But personal incredulity isn't evidence. The universe is full of things that seem implausible until you understand the mechanism. The formation of stars, the structure of atoms, the behavior of quantum particles, all seem strange or impossible at first, but evidence and explanation make them comprehensible. Evolution seems implausible only if you think of it as random chance producing complex organisms in one step. But that's not how it works. Evolution is the accumulation of small selected changes over vast time. Each step is small, each provides some advantage, and over millions of generations, the accumulation produces dramatic transformation. Given the time scales involved, billions of years, and the relentless power of selection acting on variation in huge populations, the evolution of complexity becomes not just plausible, but inevitable. Religious objections to evolution often stem from perceived conflicts with scripture, but many religious traditions have reconciled their beliefs with evolutionary science. The Catholic Church, many mainstream Protestant denominations, and numerous individual believers accept evolution as the mechanism through which God created life's diversity. They interpret their scriptures non-literally, seeing creation accounts as theological statements about God's relationship with creation, not as scientific descriptions of natural history. Science and religion address different questions. Science asks how things work, using observation and testing. Religion asks questions of meaning, purpose, and value. Evolution is firmly in the scientific realm. It's about how life diversified, not about whether that diversification has deeper meaning or purpose. One can accept evolution as scientific fact, while holding religious beliefs about ultimate meaning. Perhaps most importantly, the strength of evolutionary theory comes from positive evidence, not just the absence of disproof. Evolution isn't accepted by default, it's accepted because mountains of evidence from multiple independent sources all support it. If evolution was simply the best of several bad options, scientists would keep searching for alternatives. But evolution isn't just the best explanation, it's the only explanation that accounts for all the data. Scientists would love to disprove a major theory. That's how scientific careers are made, by overturning accepted ideas with better explanation. If there were evidence against evolution, researchers would eagerly investigate it. The fact that after 150 years of intensive research by thousands of scientists, evolution stands stronger than ever, tells us something profound about its validity. Evolution has survived every challenge, every new discovery in genetics, every new fossil, every new observation has reinforced rather than undermined it. The theory has been refined and expanded. We now understand mechanisms Darwin didn't know about, like genetic drift and horizontal gene transfer. But its core principles have proven robust. As you settle into sleep, understand this. Evolution is challenged, not because the evidence is weak, but because its implications are profound. It tells us that humans are part of the natural world, related to all other living things, products of the same processes that shaped every organism. Some find this uncomfortable, but discomfort with implications doesn't make evidence disappear. The evidence is what it is, and it overwhelmingly supports evolution. That's why scientists accept it. That's why it hasn't been disproved and won't. The evidence is simply too strong, too consistent, too comprehensive. We've looked at DNA that reveals our relationships to all living things. We've studied anatomical structures that trace back to common ancestors. We've traced the distribution of species across continents and islands. We've watched evolution happen in real time. We've seen how all these independent lines of evidence weave together into a strong, coherent picture. Evolution stands as one of the most thoroughly documented and tested theories in all of science, because the evidence supporting it is vast, diverse, and mutually reinforcing. Every new discovery adds to the picture rather than contradicting it. Every test strengthens the theory rather than weakening it. This is why evolution hasn't been disproved, because the evidence supporting it is overwhelming. More than that, evolution provides a unifying framework for understanding biology. As Theodosius Dobzhansky wrote, "Nothing in biology makes sense, except in the light of evolution." Why do organisms have the features they do? Why are species distributed as they are? Why do diseases evolve? Why does development follow particular patterns? Evolution answers these questions, and countless others, making sense of the living world's complexity. The beauty of evolution isn't just in its explanatory power, but in what it reveals about nature itself. The deep connections between all living things, the creative power of natural processes operating over vast time, the way simple mechanisms can produce extraordinary diversity and complexity. Understanding evolution means understanding ourselves as part of the grand story of life on Earth, connected to everything from the bacteria in our gut, to the trees outside our windows, to the extinct creatures whose fossils we find in ancient. Rest now in the knowledge that science at its best seeks truth through evidence and testing. Evolution exemplifies this. It's not accepted because of authority or tradition, but because the evidence demands it. And as you drift towards sleep, let that certainty bring peace. The natural world, understood through patient observation and rigorous testing, reveals patterns of profound beauty and connection. Evolution is real, the evidence is clear, and understanding this helps us see our place in the magnificent, ever-changing story of life. Thank you for listening.
If you enjoyed this journey through the evidence for evolution, please consider liking this video and subscribing to Science Lullaby for more soothing explorations of scientific understanding. Sweet dreams and good night.